Hi viewers, today we will discuss about carbohydrates classification. Carbohydrates, sometimes also known as saccharides, comes from Greek letter saccharos, which means sugar. It is classified as monosaccharides, which is basic unit of carbohydrates. When two monosaccharides combine, it form disaccharides. Combination of few monosaccharides form oligosaccharides. However, when multiple monosaccharides combine through glycosidic bond, it forms complex molecule known as polysaccharides. So the characterization of all these types based on number of monosaccharide units. And because monosaccharides and oligosaccharides are sweet in taste, soluble in water and crystalline in nature, that's why it is known as sugar. If we look at monosaccharide, it is a combination of aldehyde or keto group, carbon backbone and hydroxyl plus hydrogen atom. Change in the position of hydroxyl group around the asymmetric carbon give rise to different isomer. As you can see in this diagram, a single change in hydroxyl group position of D-glucose give rise to galactose and mannose. Similarly, change from aldehyde group to keto group form D-fructose. Then come the disaccharides. It is a combination of two monosaccharides bind together by glycosidic bond with removal of water. These monosaccharides can be same or different. Here in the first example, we can see two alpha glucose subunits form glycosidic bond when carbon 1, also known as anomeric carbon, react with fourth carbon of the other glucose molecule, it form alpha 1,4 glycosidic linkage, and the disaccharides is known as maltose. Why we called it alpha linkage? Because both the glucose subunits have hydroxyl group down the ring at anomeric carbon, so called at alpha glucose. Maltase enzyme can break down this alpha glycosidic linkage in our intestine. Similarly, combination of alpha D glucose and beta D fructose linked together by glycosidic bond between carbon number 1 of glucose and carbon number 2 of fructose. So we can say alpha 1 beta 2 glycosidic linkage are formed. Sucrose is also known as cane sugar or commercial sugar and much sweeter than other disaccharides. Third one is lactose, formed by glycosidic bond between carbon number 1 of beta D galactose and carbon number 4 of beta D glucose. Both these galactose and glucose are beta subunit, because if you can see that hydroxyl group at the anomeric carbon is upside the ring, enzyme which hydrolyze beta 1,4 linkage is known as lactase. Lactose is clinically significant because mostly infants have lactose intolerance due to deficiency of lactase enzyme, which is overcome by lactose-free milk. Finally, a point to remember about disaccharides that maltose and lactose are reducing disaccharides, because if you see, it has free aldehyde or keto group at one end. However, there is no free aldehyde or keto group at this molecule of sucrose, so it is a non-reducing disaccharides. Oligosaccharides is simply a combination of few monosaccharides It has a significant role in cell membrane, cell identification, and much more. Now come to polysaccharides. It is a complex form of carbohydrates made up of multiple monosaccharides. It could be combination of same or different monosaccharides units. Polysaccharides could be cellulose, which is plants in nature. Cellulose is made up of beta subunits of glucose forming beta 1,4 linkage. We human lack cellulase enzyme which could break this glycosidic bond. That's why human unable to digest it. Moreover, cellulose are linear chain, means there is no branching in its structure. Starch is also a predominant polysaccharides and it is also from plant source. It is made up of alpha glucose subunit either by alpha 1,4 linkage or alpha 1,6 glycosidic linkage. If there are only alpha 1,4 glycosidic bond and no branch point, then it is termed as amylose. However, if there are both alpha 1,4 glycosidic bond and alpha 1,6 glycosidic linkage and branch point, then known as amylopectin. Point to remember that at branch point there will be always alpha 1,6 glycosidic bond. Glycogen is the type of polysaccharide which come from animal source. It has same alpha 1,4 and alpha 1,6 linkage as we have seen in starches, and of course it has branching in it, but the branch point interval are different compared to amylopectin. And it is also made up of alpha glucose subunit, so starches and glycogens can easily digested by alpha amylase in human, and glycogen is instant fuel reserves in human body. Here if we look at the structure, there is alpha 1,4 linkage in linear polysaccharides, while at branch point there are alpha 1,6 glycosidic linkage. So this was all for today's tutorial. Please do subscribe and give your feedback in comment section.